Just a few decades ago, submersibles and remote sensing technologies allowed scientists to visit the farthest reaches of the ocean for the very first time. Of the many wonders they discovered, one of the most surprising was the existence of rich clusters of life flourishing in the darkness of the deep sea floor. These communities were found to be powered not by the light of the sun, but by the heat of the earth. Hello, everyone, and welcome to a new mini-series of educator professional development opportunities offered through the NOAA Office of Ocean Exploration and Research. This is the second in our new mini-series and offered by us and also our Education Alliance partners across the country. As you know, today our focus is deep sea hydrothermal vents. I'm Susan Haynes with NOAA OER, and I'm really happy to have all of you here with us today. Today, we have program participants joining us through registration from the Texas State Aquarium, from Loveland Living Planet Aquarium in Utah, from Albuquerque Biopark in New Mexico, the Great Lakes Aquarium in Minnesota, and the Dolphin Island Sea Lab in Alabama. We'd like to welcome all of the folks who are helping us from those Alliance partner sites. Can we have our wonderful coordinators pop up on the screen? I think we can start with Rosie from Texas State Aquarium. Can you say hi?
Hi, I'm Rosanna from the Texas State Aquarium.
Great, and Rosie will be managing your follow-up?
Yes, ma'am. That will be March the 17th, and you will get it.
Fantastic, thanks Rosie, so glad to have you here with us. And Kristen from Utah?
Hey everyone, I'm Kristen Smith from the Loveland Living Planet Aquarium in Utah, and I'll be sending out an email really soon with information about the follow-up.
Terrific, Kristen, thank you so much. And Alexis.
Hi, from Minnesota. I'm Alexis Berkey here at Great Lakes Aquarium in Minnesota, and I believe our follow-up session is in just about a month, almost a month to the day, so you'll get more information about that from me.
Terrific, Alexis, thank you. And Deborah?
Hi, everyone. Deborah Cook from the Albuquerque Biopark Aquarium in Albuquerque, New Mexico. So glad everyone is here. And we will, I'll see you March 10th at 4 p.m. I'll send you all the information beginning next week. Thanks.
Glad to have you here, Deborah. And Tina.
Hi everyone, I'm Tina. I'm at the Dolphin Island Sea Lab, which is not the animal, but DAUPHIN. We're down in the southern portion of Alabama, and I'll be seeing you again on our follow-up on the 16th of March at the same time.
Terrific, Tina. Thank you so much. We're so grateful to have all of these sites joining us, particularly in the Time and Society this year. So thank you all for your hard work, and we look forward to your follow-ups in the near future. I'd also like to note that this programming is possible through an incredibly significant partnership with the University of Rhode Island's Inner Space Center, and also supported by the National Marine Sanctuary Foundation. So you all received a short introductory video yesterday, and I hope you had time to watch it. And we also sent you a Mentimeter link to drop your impressions of the video into a word cloud. And I'm going to ask my magicians behind the screen if they can pop that word cloud up on the screen so everybody can see your responses. And my goodness, did we get some responses. Wow, this is fantastic. Just to read off a couple of the thoughts from various people. I see mysterious there, I see informative there, I see curious, diversity, and chemosynthesis comes up pretty large on the screen, and that will be a definite emphasis of our presentations today. I see amazing. Well, we hope that we're going to share a bunch of amazing images with you today, as well as what's in the video. And I also see Yeti crab up there a couple of times, and that is one of my favorites, and we're going to talk a little bit more about the Yeti crab a little later in the program. So thank you all for filling out that word cloud. It gives us a nice sense of of what you thought of the video. I'm glad you're excited. We are excited too to share some of the insights and the wonders of hydrothermal vents with you. So the goal of our entire series is to introduce you to experts in different ocean science disciplines and to highlight their areas of expertise, in this instance, hydrothermal vents. Also to provide you a number of different educational resources on those specific topics, and then to give you an opportunity for that collegial conversations, for those professional connections between one another and between our site partners. And as I mentioned, this is the second of several topics we're going to be presenting, so you can look for new opportunities as the year progresses. And a brief plan for our 90 minutes together here tonight. We're going to start out pretty soon with a question and answer with an amazing hydrothermal vent expert, and that'll last about 25 to 30 minutes. And then we're going to provide you with an overview of a hydrothermal vent activity that is hot off the presses, and that'll be another 25 or 30 minutes. And also share with you how to access resources from a number of different organizations that are associated with hydrothermal vents, and then a brief reminder of your follow-ups. So with that, just a little bit of background about NOAA OER and what we do and why we do it. We are a federal office that was established in 2001 with a mission to explore the ocean for national and also for international benefit, focusing on the fact that our global future depends on our understanding of the ocean. And along with federally funded exploration opportunities on a number of different NOAA vessels, sorry, I don't know how to turn that off. We also operate the NOAA ship Okeanos Explorer, which is the only federal ship dedicated to ocean exploration. And we at OER and many of our partners, like Ocean Exploration Trust, who operates the exploration vessel Nautilus, and Schmidt Ocean Institute, who operates the research vessel Falkor, all three of which you could see there on the screen, we all target areas that no one has ever been to before. And in many respects, we really have absolutely no idea what we might find. We map the seafloor, we study water chemistry, we explore the organisms that live throughout the open and also the deep ocean. And we often find things that are completely new to science and pretty incredible. And I've got a couple of things that I wanted to share with you just to whet your appetite for the things we're going to be talking about today. First, I believe we have a short clip of a hydrothermal vent system in the Marianas, and this was found in 2016, towering 30 meters tall. That's as tall as a 7-story building. And this is the chimney system that was highlighted in as part of the Deep Sea Dialogues video that you saw yesterday. Another thing I wanted to share with you that's just fascinating are these incredible snails that explorers found at vent chimneys specific to the Indian Ocean. And these snails, if you can see that snail in the bottom left-hand corner, it's got scales all over its foot. And these snails are found at a depth of over 2,500 meters. And the unusual foot is actually armored with small iron mineralized plates, we presume for protection. And just as interesting for the snail, its heart comprises about 4% of its body volume. So that's pretty significant. That's a rather large heart for the size of that animal. And lastly, the last one I wanted to share with you, and clearly you guys are excited about this too, is one of my favorite animals. It's called a Yeti crab. It's a very hairy crab, and it actually farms bacteria on its body. And I'm going to leave that right there with hopes that our expert that I'll introduce you to in a moment will share a little bit more with us about the interesting adaptations of the Yeti crab. So there's always something new to learn when we're exploring the deep sea, and it's certainly one of the reasons I love my job. And as you saw in the Deep Sea Dialogues video that we shared with you with Dr. Flinders and Dr. Aman, These vents are incredibly dynamic, incredibly volatile environments, and they're teeming with life at the same time. The vents are homes to diverse ecosystems that actually exist in the complete absence of light. And bacteria, instead of plants, are the base of the vent food webs, gathering their energy from the chemicals in the vent fluids to produce the simple sugars through the process of chemosynthesis, which we'll also explore and talk about a little bit more later. I just wanted to make a note of the resources that we'll be providing. There will be a huge stack of resources for you in a Google folder, but you're also going to find an introduction to the ships that I mentioned, the ships of exploration and our strategies for exploration. You're going to find a number of different activities, fact sheets, essays, and a whole lot more. So I hope you look forward to digging through those materials for things that you can use in your classroom. So all that said, I hope you all arrived with good questions for our guest vent expert. And I'd like to introduce you to Dr. Peter Gerges. And Pete is a professor of evolutionary biology at Harvard University. He's also an adjunct scientist at Woods Hole Oceanographic Institution. He carries degrees from UCLA and UC Santa Barbara and did his postdoctoral work at Monterey Bay Aquarium Research Institute. In his lab at Harvard, where he's been since 2005, he and his team study the physiology and the biochemistry of deep sea microorganisms. So they look at the physiological relationships like nutrient exchange and immune responses between microbes and animals in the deep sea environment. And additionally, he and his team like to build a variety of tools to do work down in the deep sea. So Pete, hi, thanks so much for being with us today.
Hi, Susan, and hi, everybody. It's really wonderful to see you all. It's great to be here. Thanks for the invite.
So glad to have you. So our participants, I'd like to ask you all to share some questions with us in the chat box. And we're going to be managing those and selecting those throughout the next half hour or so. But I'd like to just start off. Pete, you do some pretty unusual and exciting work studying both the large and the very, very small organisms of the deep sea. Can you briefly talk to us a little bit about what got you interested in this work?
Yeah, that's a great question, because it's an opportunity to remind all of us that our journeys from being kids to being, adults are not always as direct as we might think, especially as kids. So I grew up in Los Angeles in a town called Downey, and Downey was a home of a bunch of aerospace companies. And while I was a kid, I loved the ocean and I loved rockets. Here's a picture of me with my parents, who are immigrants from Egypt, by the way. And this is in front of a water ride at Disneyland I was just obsessed with, right? So my love of all things water came at an early age, and my love of all things exploration. And I think that it kind of got baked into me, this idea that you could sort of spend your life studying the world around us. Now, as a kid, I didn't know that I could pursue a career in this, but I knew that I loved it. And I just found myself always drawn to the idea of studying the world around us.
Fantastic, fantastic. And it's interesting you mentioned Waterworld. It's just a testament to those informal education places, right, across the country that can inspire young people to do some of the most amazing things in the environmental world.
Absolutely.
So, it's great. And it's a great picture of you when you were a child. When we met last week, you also described yourself and those in your lab as widget builders. Can you tell us a little bit about how that fits into the context of your work?
Oh my goodness, the lab folks, if they hear this, they're going to be like, Peter, what are you doing? What do I mean by widget builders? So, I was just telling you about how I grew up as a kid in this town where literally my neighbors were employed at these companies that built the rockets that went to the moon. And they built the space shuttle, literally. And it took all of them working together, right? From engineers to machinists to the custodial staff to human resources. It was a team. And that was very clear in this little town of ours. Today, as a scientist who loves and cares and wants to study and learn about the deep sea, it occurs to us that to do that, we're best served by kind of taking advantage of each other's strengths and working as a team, and building technologies that help us ask questions that we might not otherwise be able to ask. So for example, if you sort of stop for a second and remember that the deep sea is cold and dark and there's a lot of pressure and it's very salty, and that means things will crush, metals will corrode. It's not easy to work down there. But what we do in the lab, myself and the other members of the lab, is we start by finding a science question that thrills us. How do we, for example, understand the microbes that eat all the hydrogen coming out of a hydrothermal vent? How do you do that? Well, to do that, you have to study the microbes and the hydrogen. So we find ourselves building and adapting a lot of tools to make those measurements of hydrogen, for example, right? Which is a gas and it's dissolved in the water. So we have to get it out of the water, and we have to measure it, calculate its concentration. Then we have to find the microbes. So that's the widget building. It's finding a question and kind of being willing to stick your neck out a bit and try to build something to help answer it.
So it's really a lot of interdisciplinary problem solving, right?
Yep. And as a kid, I completely remember thinking like, why do I need to learn this? Like, it doesn't make any sense. Or why do I need to learn that? And as I got a little bit older at a time, I got to see how those things connected. And so while I don't, use the heavy duty math that I learned every single day, it does pop up. And understanding it helps me design and build these really cool tools to study the deep sea.
Fantastic. That's a great inspiration for our educators that are constantly helping students to make those connections. Thanks.
You're welcome.
So we have magicians behind the screen who are helping us manage questions, and I think they're going to start popping up. So Casey has asked, how far away in general is life established from the vents? So do you find species that have separated into zones in relationship to the hydrothermal vents?
Thanks for the question, Casey. So let's zoom out, if you will, and talk about life in the deep sea. We now know that life exists everywhere in the deep sea, right? So it's not as though life is only found at vents. It's also, there's plenty of life away from vents. If you talk about the animals and microbes that are found in vents, they do kind of zone around those vents. So for example, we find some of the tube worms that live around hydrothermal vents stuck in these kind of areas where the really hot water mixes with the cold water. And Casey makes kind of bathtub temperature water, like literally bathtub temperature. That's where we find a lot of the worms. Some of the snails you see on the screen now, they live in slightly warmer waters. A lot of the crabs you see on the screen now will even wander into even warmer waters and pick at the microbes. But the zonation is an important part of the vent ecosystem. That is, in essence, their ecology, Casey. So other scientists, besides myself, spend a lot of time thinking about the zonation and why it exists and how it remains in balance. It's a great question, Casey.
That's interesting, because you think about zonation in any ecosystem, in any environment, right? Whether it's a seamount or a tree or an intertidal area, and it's true for hydrothermal vents too.
Absolutely.
Oh, great. Can we pop up another question, folks? Matthew asks, he's curious about the evolution of the organisms since they live in such remote and actually fragile areas.
Yeah, thanks, Matthew. So when people first discovered the hydrothermal vents at the end of the 1970s and the beginning of the 1980s, there was a lot of many questions about where did these animals come from? And Matthew, one of the first ideas was that they were actually very ancient organisms, not just the microbes, but maybe even the animals. Since then, especially through DNA sequencing, we've been able to show that a lot of them, Matthew, are actually related to animals that live in shallow environments. So some of the crabs that we find at vents are actually more closely related to those in the intertidal than they might be another deep sea crab. So to answer your question, Matthew, they are kind of remote. The environments are, we'll use fragile, but let's say that they're dynamic. How's that? The cool thing is that the animals we see there are not just static. That's not the same assemblage of animals that's been there for 100 million years. Animals have come and gone. And so it's over evolutionary time, it changes. So what we see now is a snapshot of the animals that have been there. And to answer your question about remoteness, believe it or not, the animals we find at one vent, say off the coast of Mexico, many of those animals are found at vents off of Easter Island. So it tells us that they're connected in ways that we don't yet fully understand. So thanks, it's a great question.
And David asks us, he was curious about stratification also. Do the vent communities look different as you move from the top to the bottom?
Yeah, thank you, David. Sometimes they do. Some of the sites we study in the South Pacific have two different kinds of snails or more and have mussels. And there's this weird thing that happens where we see some snails way up at the top, kind of at the tippity top of those spires, of those chimneys. And we find some of them kind of in the middle and others along the bottom. So it's a great question. This was actually a big part of my colleague, Professor Chuck Fisher's research about 4 years ago when we were there. And I think that the students and postdocs who are working with him are still trying to look at the patterns and figure out, why are some of these hairy snails you see here called Alphinaconcha, why do they creep up to the top? And weirdly, why do their shells look a little different? They're a little different in color and so on. So we're still trying to unpack all of that.
Could some of that snail color have to do with the minerals that are in the water?
Almost certainly. But it's still strange because the snails are mobile. So if the colors are different because there's minerals sort of settling on them, how long have they been there? And why aren't they moving around? Maybe they don't move as much as we thought. So there's a lot of questions that we can't answer because we can't just walk down there and check them out every day.
Right, unfortunately.
Right.
It's amazing what we don't know. Right?
Isn't it?
Christine asks, what adaptations do organisms have to deal with that extreme heat?
Oh, Christine, that's a question that's near and dear to my heart. It turns out that the tolerance to heat that we see among animals is very different than the tolerance to heat that we see among microbes. Let's start with the microbes. We find microbes living around vents at temperatures of about 122 degrees Celsius. That's pretty darn warm, and it's about the temperature you might, start to bake things in your oven at, just to kind of put it in context there. That and the microbes survive that because they have evolved to tolerate those temperatures. But it's important to remember, Christine, that the water's not boiling. It's hot, but the pressure keeps it from boiling. Boiling is really hard to live in because water turns into a vapor and pops cells really well. So somehow they have figured out or evolution has figured out how to get them so that they can live at those high temperatures. Animals, though, which all have mitochondria, don't really do well above about 60 degrees Celsius, or about half the temperature. And it looks like a big part of the problem is that the mitochondria, again, upon which all animals are dependent, don't seem to function well at those high temperatures. So that's about the upper temperature limit of animal life as we know it. It may change a bit, but that's where we're at now.
Wow, that's amazing. That's absolutely amazing. What are some of the major challenges of studying the vents? The folks at the Village School Middle School, they said it seems super dangerous, but pretty thrilling.
For all you Village School, Middle School folk out there, what are some of the challenges? Well, I would say that the biggest challenge of studying vents is that the environment itself is really dynamic, and it changes really quickly over really short spatial scales. Let me give you an example. If I go to the desert out in Southern California where I grew up and I want to measure the temperature, I can pretty much take the air temperature or measure the temperature of a rock or under a bush and know that the temperature is going to be not totally uniform, but it's not going to change that much. But check this out, folks. At a vent, there will be a spot on the surface of a chimney that's 120 or 130 or 150 degrees Celsius. There's water that's 350 degrees Celsius. And just a few centimeters, just a couple inches away is ice cold water. And it's totally dynamic. It's really hard to measure that on land, let alone in the water. So the real challenge, folks, at least in my opinion, for the kind of work we do, is that we go down with this, I forget how heavy the submersibles are, but it's many, many tons of a submersible or robot using these kind of clunky-ish manipulator arms, holding a temperature probe that's, a foot and a half or, almost, half a meter long. And we're trying to measure a temperature while everything's sort of moving around us. So just the nature of the environment actually makes it hard to do some of the kind of work we really want to.
Yeah, the skill and the talent to do it is interesting also, right? The ROV pilots who are managing all of those tools. And
yeah, and if you don't mind, Susan, I want to give a shout out not only to the ROV pilots, but to the entire ship's crew. I would argue that when I go out as chief scientist, I'm probably the least important person on the boat, or the least useful, because the ROV pilots have to fly the vehicle. The people on the ship's bridge have to keep the ship from wandering around. The folks in the engine room have to keep the engine running so the ship doesn't just take off anywhere. So this huge team all contributes to doing that science, and we frankly just have the privilege of benefiting from it.
Yeah. It takes a village, right?
It takes a village.
Getting into a little bit more about some of these organisms, and Cassie has asked about the microbes chemosynthesizing for tube worms, and are they found in other animals like oysters or clams or?
Thanks. I guess I'll call you Cassie W2. We do know that there are these kinds of chemosynthesizing microbes in other organisms. Let me take a step back and just remind all of us what chemosynthesis is very quickly. Every living thing on Earth has to harness or get energy from outside of itself. That is a fact. So think of ourselves as a kind of a biological version of a car. A car, you have to fill up the gas tank with the gasoline, that's its food, and it uses oxygen to combust it to do work. As a human, you eat stuff and use oxygen to, not combust it exactly, but oxidize it. That's a kind of chemical way of getting the energy out of the food you eat. Plants get their energy from the sun. Chemosynthesis refers to microbes and processes that get their energy out of chemicals. And when we talk about it at vents, we're usually talking about microbes that take chemicals that come out of the vents. Something like hydrogen sulfide, that rotten egg smell, that you might catch a whiff of on a sewer. You can actually take that and use it, react it with oxygen and harness energy from that. That's what chemosynthesis is, right? That's all it is using chemicals for energy instead of the sun. Okay. Now, back to your question. We know that these kinds of chemosynthetic microbes form partnerships with a bunch of different kinds of worms and a bunch of different kinds of bivalves. Those are things like mussels and clams. I don't know of an oyster yet. It's an interesting point. But we also know that they'll form other associations. We call it like epibionts growing on the outside. There'll be epibionts of all sorts of different kind of organisms. We see some on sponges and we see some on other kinds of worms. So it's much more diverse than we thought originally. It's a wonderful question.
So to spin off of that a little bit, can we go to that amazing Yeti crab? What makes them so interesting?
So I have two questions, two responses to your question. What makes them so interesting? Answer number one, I do not know. Look, I've been studying these fin animals for a long time, and I love the Riftia tube worms, the big giant tube worms that live at vents. They have no mouth. They don't have a gut. They don't eat anything. And they grow billions, if not trillions of bacteria deep inside them, right? So to me, that blows my mind.
Right.
So I don't really know. But the second thing that's cool about Yeti crabs that I see and sort of understand is that there are these weird crabs that got this fur on them. What is that all about? Here's the deal, folks. Yeti crabs are farmers. And a lot of animals that live around vents are also farmers. They have evolved the ability to grow microbes on them. And it is a kind of farming. They figured this out well before humans did. So they spend a lot of time making sure that their farms, their bacterial farms, right? That's what you're looking at, this fur, it's bacterial farms, that they spend time moving around, giving the bacteria access to that hydrogen sulfide I mentioned, giving them access to oxygen, and making sure that they live. And then we're, presuming that they feed on these. Like maybe they graze on the one on their neighbor's back, maybe they graze on their own arms. I'm not so sure of that. Others may have a better idea, but they're farmers, and that's pretty darn cool.
Farming the hair on their body. How
Right.
unusual.
Yeah.
And don't different Yeti crabs live specifically on different vents, just
Yeah,
like some other organisms?
Yeah, they do. And some of them, like you just saw, are kind of sitting there. Others are sort of waving their arms in the air, and that's just so cool to watch. So yeah, imagine growing, microgreens on your arms and just kind of picking them up and gnawing on them. That's pretty wild.
That's pretty crazy. Yeah. Along those same ideas of organisms, Danielle had asked if the shell of the scalyfoot snail is calcium carbonate or if it's iron. And if it's not iron, how does it survive in the lower pH of that area?
Ooh, well, Alexandra, you're asking a real kind of deep chemistry question there. So let's start about snails and these kind of calcium carbonate shells. A lot of organisms build shells out of calcium carbonates. And to my knowledge, so I'm not a scalyfoot snail expert, their shells are, like all others, calcium carbonate. What we see in terms of the scales and the discoloration on the cell is the discoloration of the shells is the deposition of iron-bearing minerals, very much dominated by iron sulfides. Now, iron sulfides, one of them is pyrite. And pyrite, folks, is fool's gold. So you may occasionally catch a glimpse of something really gold and sparkly at a vent, and it's probably pyrite or versions of pyrite. The scalyfoot snails, I think they have a precipitate around them. And, Alexander's question is an important one. How do they keep them from dissolving? Not knowing the specifics about the snail, let me just tell you that for a lot of the organisms that live in these lower pH environments, I suspect their shells are dissolving. But this is a high energy environment. There's lots of energy at vents. And with all that energy and all that food, I suspect that they're constantly renewing and investing and rebuilding the shell portions that may dissolve at these different conditions. But remember too, they're not all living at really, really acidic water only. A lot of them move in between a more seawater-like water and the more acidic stuff. So
right, that's interesting because there's been a lot of conversation about the acidification down in that area, and that's a great point that a lot of these organisms don't live right there where it's quite so toxic, right?
Yeah.
And I love the concept of, wow, well, there's so much energy happening there that the regrowth can be happening at the same time.
Yeah, and that's a big difference, folks, between vents and the rest of the deep sea, is that they're high energy, lots of food, and they're very physiologically taxing. But at the end of the day, what matters is that you have enough energy to cope with that and grow and reproduce and have babies. If you can do that, then you do it, right?
Serious survival of the fittest.
Yep.
Great. Do we have some more questions to toss to Peter? We just asked that question from Danielle. We just
Thank
spoke
you, Danielle.
Yeah. While we're waiting for another question, Peter, I'm going to ask you. I know there's some differences between how hydrothermal vents form and the activity in the Pacific and the Atlantic. Can you talk a little bit about that and maybe why there's some differences and what those are?
Yeah, that's another important question. So let me give you the short answer and then unpack it a bit. Not all vents are the same. There's lots of different geological settings in which we see venting. And there are some general trends depending on the ocean basin you're in. So in the Atlantic, we have a lot of slower sort of spreading ridges, and the vents seem to be longer lived. In the Pacific, we have some faster sites, and the vents tend to be shorter lived. And then, of course, there are other different kinds of geological settings, things called back arc basins, where we see vents as well. That's the broad brushstrokes. Here's what's cool, though. my colleagues I mentioned before, Chuck Fisher, another professor named George Luther, Roxanne Beinert, who I believe you'll be talking with soon, among many others I'm sure I'm forgetting, have done quite a bit of work down in the South Pacific at a place called the Lao Spreading Center. And at those vents, we have been trying to understand the ecology and physiology of the animals. One of my favorite stories is when Professor Fisher and his students told us about how they wanted to see if the snails, if they moved them from one spot to another spot, if they would come back because they're mobile. And that was our original hypothesis. We thought, wow, if you're mobile and you live at a vent, how cool is that? Because if the vent changes, right, if the flow regime changes, you just go. Unlike a tube worm, you're stuck where you're at. So they would pick these snails up, move them, come back a day or two later, and nobody will have left where they put them, or they'll be gone and they won't have returned to their original spot. So there's a lot about that was confusing until we looked at a lot of the chemistry and realized that this vent in the South Pacific was remarkably stable. From a chemical point of view, not much has changed. And when they went back and looked at time series of where animals are located, it's as though nobody moved over 9 or 10 or 12 years. So we're still trying to understand why that system seems so stable and others in the Pacific aren't. There's just a lot we still don't understand. So a lot of big differences among vents.
Wow, and so much of it is dependent on that geology and understanding the geology as well as the chemistry and the biology, right?
Spot on. You cannot ignore one and just study the other.
Yeah. Thank you for that. Emily has asked us, have we started mapping or tracking vent systems to see how they change over time? That's a good question.
Yes, we have, Emily. That's a wonderful question. There are a number of vents that have been study sites for a couple of decades now, and people go back again and again and again and look at the evolution of those vent systems. Probably the best studied one is on the East Pacific Rise. It's at 9 degrees north latitude, and so it's often called 9 North. And if you're interested, Emily, you can find a lot of papers, including some in the kind of more popular literature that talks about life and death and succession at these vents. So we're starting to get a grasp on it.
Is that the one near the Galapagos?
Yeah, it is. Yeah. Absolutely.
Yeah.
And even the one at the Galapagos has changed over time. The folks at the Ocean Exploration Trust, in collaboration with NOAA, went out not too long ago back to the original vents that were sort of first observed, and most of them are gone. And a whole bunch of other new vents have popped up not too far away.
Yeah. Can you talk a little bit about succession in relationship to that?
Sure. So one of the more interesting things about vents is that they are, as you might imagine, cracks or fissures in the crust, and they're kind of a bit like a coffee percolator. Now that's going to make sense to some of us who have gray whiskers, but hot water comes up through these cracks into the ice cold water, and this smoke you're seeing, folks, is not smoke. It is more or less metal dust. And you can imagine that as that metal dust comes out and it sticks together, it forms an amalgam. That's what a chimney is. That big, tall sulfide behind there, it's a bunch of metal dust that's more or less stuck together, through chemical reactions and other processes. But you can also imagine that dust causes the orifice or the opening to seal up. So vents have a lifetime that has to do with many, many sort of dynamics of the vent flow and the temperature and the chemistry and so on. Succession happens as vents change. So if there's an earthquake, for example, or an eruption, we see new vents arise and we see colonizers show up. Lots of microbes at first, lots of microbes. And little by little, we start to see the presence of certain kinds of animals, some tube worms, some crabs and other things start to show up. And as the community matures, you see some of those original sort of founders, sort of colonizers die off in abundance and other species come up. So vent succession is pretty wild, and it's different all around the world, right? What we see in one spot is not the same as another vent.
Interesting. Victoria had asked, how big are the direct impacts to the environment surrounding the vents? You talked about all of that, the black minerals coming out. How far out do the minerals actually spread from the source?
I love that question. Victoria, I would say, we have not done as much work as some of us would have hoped. That metal dust, and some of the sort of other kind of chemical components often go up above vents and form what's called a buoyant plume. So imagine this hot water comes billowing out and it gets to a level where it's just cool enough and its density matches that of the surrounding seawater, and it just sits there, right? And sometimes some of that comes back down and it spreads all over the place. But if you look along a mid-ocean ridge, the mountain chain on the seafloor, and you look at the sediments around them, they are enriched in metals. They're often called metalliferous sediments. That's a fancy word for saying that there's a lot of this metal dust. And I'm very interested in that as well as others and members of my lab in understanding who's living on that metal dust. Colleagues of mine, I'll mention one, Professor Jason Sylvan in Texas, does a lot of research on studying microbes that live off these old extinct sulfides even that are far away from the vents. So it's a wonderful question, Victoria. There are probably whole ecosystems and communities built around that.
And Emily asked, are they able to age the vents based on the metals that make them?
They are able to age vents based on radioisotopes and their decay. So while our knee-jerk reaction as humans is to think, oh, radioactivity, there's a lot of naturally occurring radioactivity, and it actually allows us to more or less timestamp with some precision, depending on what you're talking about, to kind of put an age on these vents. So there is quite a bit of research that's been done on looking at how old vents are, how old some of the mineral deposits are. And remember, the sort of sulfides themselves are quite young. But a lot of the extinct sulfides, there's ongoing efforts on aging them to see how long they last.
Fantastic. Jordan has asked us, how has chemical change affected these vents? I'm sorry, how has climate change affected these vents and the creatures that live there? Is that an impact?
I don't, it's a great question, and I don't know of a huge body of research on that yet. When we think about climate change, we are really, in the ocean, we're really talking about increases in temperature and reductions in pH. It just so happens that the vent animals like those you're seeing here, this is a vent shrimp that can kind of navigate using thermal detection in and around the vents to feed on microbes. Super cool shrimp. They're pretty well adapted to dealing with changes in pH. And the changes that we're going to see throughout the ocean are fairly modest. So with my physiologist hat on, my hypothesis would be that increasing changes in ocean temperature and pH are not going to affect the adults. Will they affect the juveniles or the larvae? That's a whole different question. Will they affect the rest of the deep sea? Almost certainly, without exception, in a really bad way.
And do we know how these juveniles and larvae travel around and spread? Do they spread from vent to vent?
We're getting a little bit better at understanding that they do go from vent to vent. I mentioned a little bit earlier that tube worms, for example, that live in off the coast of Central America and Mexico, they show signs of, we'll call it population mixing with the tube worms down off Easter Island. So that tells us that we would call it gene flow, right, across that ridge crest. The big question is, how many vent have larvae that sort of float up way high into the water column and then rain down like dust? That seems really hard to believe for many organisms because the odds of you settling on a vent are lower. Now, some organisms have larvae that have lots of, energy stores like yolks and things like that. But there are many others that we think just travel along the ridge axis. Does that make sense? So if there's a current just flowing along this mid-ocean ridge system, which in times is kind of like a valley, you could imagine these currents taking, larvae from one site to the next, kind of hopscotching their way around. Right. That's probably a more common approach.
Sort of similar to what might be happening with seamounts?
Completely. Great analogy. Great analogy.
Okay. Pete, we have just a couple more questions for you. And this is one that, you all get as scientists, and it's an important one from an educator standpoint. Holly has asked, being a family of immigrants yourself, do you have any advice for students who are also first-generation Americans and are pursuing a STEM career?
Yeah. Thank you, Holly, for asking that. There's a lot to unpack there. And I would leave you with the following sort of key points. First things first. At the end of the day, I would encourage you to pursue a career that brings you joy, something that really thrills you, and to worry a little bit less about to worry a little bit less about whether or not you're going to be successful at it at the beginning, right? Do your work, etc. But if you love it, you're gonna work at it. You're gonna be excited to do it. Bear in mind that if you are interested in the ocean, but you have a love of finances, that is actually okay. So the second point I want to make is there's a lot more ways to engage with something you love other than strictly just being that. You don't have to be a marine biologist. The ocean needs people who are good at financing, at weather modeling, at people who are good at engineering. The ocean needs people who know how to run ships and so on. So think broadly about what makes you passionate, how you can contribute, and finally, find the people who are going to support and nurture you. It is hard to make our way in this world without having a kind of network. And I, at a young age, my parents, frankly, didn't know academics in California. We didn't have friends who sort of knew how to navigate the university system. And I'm so lucky, folks. My 5th grade teacher, Mrs. Rucker, Mrs. Tadakoro in 10th grade, and Mr. Hamner, or Professor Hamner, and Professor Muscatine, and Professor Chapman. Those 4 people made a huge difference in my life in my early days because they helped me out immensely. So find people who are your allies and are going to help you get to where you want to go.
Mentors and inspirators, right? Both so important. And as a slew of educators across the country talking with us tonight, it's so important for an educator to be an inspirer, right?
Absolutely.
And it sounds like you had many of those in your life, which is
absolutely. Major shout out to the teachers and educators, museum curators, and all of you folks. You are more inspiring than you realize.
Yeah.
So thank you.
Yeah, that's great. That's great. I think we have one last question that our team has pulled up. Thinking a bit about food chains and how those food chains work. Are there animals out there that eat things like Yeti crabs? What does the food chain food web look like down there?
So 2 answers. First is there is a food web. Microbes are at the base of that food web. That is absolutely the deal. The overwhelming majority of primary production occurs by microbes. Now that's just a fancy way of saying the animals that harness energy from things that aren't organic in nature and turn it into organic matter, right? Plants take sunlight and carbon dioxide to make sugars. At vents, the microbes take the chemicals, hydrogen sulfide and oxygen and carbon dioxide to make sugars. So they're the primary producers. They're the functional equivalent. They're the equivalent of plants in the deep sea. Lots of things graze on them. Limpets and scale worms and all sorts of other, little things munch on them. And of course, things hunt those and eat those. That's the first answer. So the second answer is, I don't know. And I wanted to share that just to remind you all that I'm an expert on a lot of this stuff, there is no one sort of wise old person who knows everything. So I wanted to turn that around and ask you all, if someone wants to figure out what eats Yeti crabs, I think that would be very cool because I have no idea. So if anyone listening wants to figure it out, and I'd love to get an email. I'm not that hard to find. That would make my day. So.
That's great, Peter. We're one of the activities that we're going to be talking about tonight is our food webs and food chains, and finding a hydrothermal vent food web is not an easy thing to identify, right? So no. There aren't that many that we really understand.
That's right. Yeah.
Yeah. Great. Well, Peter, you are an inspiration yourself. Just having you share your expertise and your incredible enthusiasm for what you do is so fantastic. Teachers don't get to talk to people like you very often, and it's such a pleasure to have you here tonight. Would you be willing to stick around and answer a few more questions a little later in the evening?
Are you kidding? This is the good stuff. I'm happy to be here.
Terrific. Thank you so much. And thank you for leading directly into the conversation that we're going to have about this activity. So we'll see you a little bit later.
You bet. Be well.
Great. So everyone, I hope that was inspiring. I'm sure it was. It definitely was to me. And I'd like to shift now into some of the hydrothermal vent instructional resources that we have available to help you cultivate your students' interests in these ecosystems. And you will have a package of resources that we'll be sharing with you, and we've shared a few of those with you already. You'll have an extensive vent resource list that highlights resources from multiple different organizations. And you also do have right now in your hands a draft activity, hot off the presses, called Life on a Hydrothermal Vent. And I'm going to ask that you make sure you have access to that this evening. You also will receive fact sheets simple fact sheets for your students on hydrothermal vents and chemosynthesis, and a few exploration notes that are first-person accounts from a couple of different scientists who explore these environments. One is from Diva Aman and Deb Glickson about the unique aspects of vent ecosystems, and those are the two of the folks that were in your Deep Sea Dialogues video. And also an essay about tracking down hydrothermal vents. How do we sniff them out from a chemistry standpoint and a technology standpoint by Dr. Ed Baker. So with that, let's move into the activity itself. This activity is an investigation, and as I mentioned, called Life on a Hydrothermal Vent. It is a high school level activity that was developed in cooperation with NSTA and with some next generation science standard experts. And we approached this differently than we've approached our NOAA OER activities in the past, with a focus on modern instructional pedagogy and a strong eye towards student sense making. And I think perhaps most of you have heard that phrase before, but some of you may not be familiar exactly with what it means, and we're all still learning. This is an instructional approach where the students are actively trying to figure out the science concept or to understand a presented phenomenon. And they're doing that by sharing their ideas, evaluating competing ideas, giving and receiving critique from one another, and also reaching group consensus. And you saw the image that just got pulled up on the screen a moment ago. Our NSTA colleagues used the Open Sci-Ed instructional model as the basis for this activity organization. And there's a link to that whole piece in your resources. So you'll have some background information to think about that more later. So here we see that the students are essentially taking a journey of discovery and understanding rather than being told and then regurgitating. So our NSTA colleagues first built this activity out as what they call a daily do, which has become a very popular new package of sense-making activities that NSTA is providing to teachers and parents to engage students in authentic and relevant science learning focused on specific phenomena and science and engineering concepts. Again, I know this is rather new to the teaching community. It's also new to us. So we will be very interested in your feedback to this approach and hope that you will bring some good conversation and thought to your follow-ups in the next couple of weeks. So if you will reference your activity handout, Life on a Hydrothermal Vent, I'm just going to kind of go through the pieces of it and guide you through the student understanding. Pages 1 and 2 are a simple overview, and the focus of the activity is to explore the phenomenon, how can ecosystems survive without sunlight? And also for the students to develop their understanding of chemosynthetic ecosystems and the dynamics through sense making. And you'll see on that first page that there are a number of different objectives. And when you actually look at your handout, you'll see that the objectives are in different colors, and they're color coded by the next generation science standards 3 dimensions as they're color coded in the NGSS book. So the objectives are for students to ask questions to investigate what causes dynamic ecosystems to survive in the absence of light, and also to identify patterns in ecosystems to develop a model to explain how the components in an ecosystem interact in the absence of light. So you can see all the 3-dimensional pieces that are in those objectives, and we've listed the specific next generation science standards that are supported by this activity. So again, the driving question or phenomenon is, how can these ecosystems survive without sunlight? And possible guiding questions are provided throughout the activity. And these are student-generated questions that come up based on student observations of the phenomenon. And these questions can be used to engage students in the process of sense-making as you move the activity and their learning forward. And I will note that on the bottom of page 2 of your handout, of your draft lesson, it references a one-pager, and that doesn't exist yet. Instead, we have provided the Open Science Ed link for your reference. And so just to orient you to the activity itself, the entire package, there are layers to this activity that guide towards student sense making. So there's an introduction to the phenomenon. And in that portion, they're engaging in observation, and they're also developing common questions and ideas. And then they move on to figuring things out, and then to synthesizing their thoughts, and then to putting all of the pieces together. So on page 3, we start with the introduction to the phenomena and engaging the students in observation. And so we're asking the students to simply think about and record what they know about ecosystems, including food chains and food webs, and interactions between the organisms in their environment. And I really want to note that throughout the entire activity, the instructor is encouraged to promote student thinking both individually and with a partner, and shared with the class, with the idea here that we are really trying to emphasize student equity and ensure that all the students' voices are heard in a classroom, whether they're the loud voices or the quiet voices. So next, as we move on, the students are introduced to the phenomena by observing these 4 images of a hydrothermal vent ecosystem in the Marianas. So we're going to show you these 4 images just to get students starting to think about hydrothermal vent systems. And these are near the Marianas Trench in the Western Pacific at a convergent plate boundary. And in your activity, you also have a false color map of the specifics of where these images lie in the Pacific. And as the students are looking at these different images, we ask them to record their observations and discuss and compare them with a partner. And then once we've done that, and you can see the map here of the region here, and you can see the Marianas volcanic arc, the red area in the center, which is a primary location for the vent systems. So once the students have observed those images and the map, we introduce a hydrothermal vent video, and this video is from the 2016 Marianas expedition by the NOAA Office of Ocean Exploration and Research. And the students, while they're watching this, can record their observations. It's a rather short video. It's about a minute, and I'd like to have you watch here. And as you're watching, consider what things your students might notice. So can we pull that video up?
So right now we are at a depth of 3,275 meters, looking at an incredible black smoker hydrothermal vent chimney. That black fluid coming out there is likely between 300 and 400 degrees centigrade. These ecosystems are literally islands or oases amongst what is mean, it's really cliche, and the deep sea is definitely not a desert, but relatively, islands of abundant fauna, islands of life in what is a usually desert-like deep sea flow. Oh, this is so cool. What
About 27 meters high, 30 meters high.
So that was the 30-meter tall vent system that they found in 2016 that they were referencing in the Deep Sea Dialogues video. And so after the students observed this, you can discuss the depth where the vent system was found, which was 3,275 meters, or 2 miles under the sea surface. And using the image on page 4, you can illustrate that distance for sunlight doesn't travel that far in the ocean, and just emphasize that it's deep and dark down there. And then we also move into a second vent system that's in the Galapagos. This is a black smoker hydrothermal vent that the exploration vessel Nautilus found with the Ocean Exploration Trust team. And this one is 10 meters tall, billowing out dark superheated fluid. And I'm just going to show you a 30-second clip from this 4-minute video just to give you a taste.
It does, doesn't it?
It is a beehive.
Wow.
How long have we known about when was the first vent discovered?
The first vents were discovered in '77. The first
black smokers.
First smokers in '82. That crab on the right
look at that.
may be Predator.
That's pretty cool, right?
Cyanogrea.
So just to give you a sense of the excitement for that black smoker video, again, having the students record their observations here and any new questions that they might have from that 4-minute piece. And then moving on in the process to page 5, the students start to develop common questions and ideas. And working in small groups, they identify their top questions and why they elevated those to importance. And those questions, as their thought processes are evolving, can include things like, are there hydrothermal vents throughout the ocean? What's the black stuff coming out of the vents? How do they live there where it's so hot? And what do they eat? And how do they get their food? So in the vein of student sense making, we can ask the students if they want to what they want to figure out, and use their curiosity to drive the direction of the lesson. And it's important to note here that some of your students are going to have some misconceptions, and there's no need to correct any misconceptions at this point, because the whole idea is for them to figure out things as they progress through the activity. And speaking of figuring out, on page 6, we move into having the students brainstorm how they think life could exist and survive at a hydrothermal vent without sunlight. So you can listen for groups to discuss different ideas that are listed there in your activity. One of which is, it must be that something besides plants and algae must be the producers of this food chain, food web down here. So at this point, progressing through the activity, the students have likely figured out that there's organisms that live in these deep, extremely hot environments, that these vent communities live near tectonic plates, tectonic plate boundaries, and that sunlight doesn't reach that deep. So the food webs have to start with something other than plants and algae. So here we provided you with an opportunity for students to discover vent food webs, and I mentioned to you that there aren't that many that we know enough to actually build a food web. So on pages 12 and 13, we've provided you with one particular web as a student activity sheet, and then also a complete food web answer sheet. And that's the answer sheet there. And you can see the variety of links from primary producers all the way to top order carnivores. And you can also see that, that this entire food web is starting from the simple chemicals on the far left-hand side. So we are providing you with a PowerPoint in your package with all of the pieces in order to work with to do this activity online if you'd like to. So there are creature cards that go along with that entire food web activity. So after the students have completed their food web, they should be able to note that there are likely a lot more organisms living at hydrothermal vents than they might have thought. And they're probably deducing that the food web seems to start with chemicals and that bacteria are the beginning of this food chain and food web, that they are the primary producers. So then we can have the students again work in small groups to kind of think about an explanation of the interactions between these organisms in the vent system. And their new questions might include things like, how do bacteria live at the vents? The bacteria that are living at the vents make their own food. And what's the process where energy is being created without sunlight? So now you've got the students thinking and they're being led down a path. On page 7, you can begin with that comparison between photosynthesis and chemosynthesis that Peter was talking about earlier today. So we can start by guiding students through the inputs and the outputs of the process of photosynthesis, and the inputs being carbon dioxide and water and energy from the sun producing glucose and oxygen. So we can have students record the things that they notice about this equation and discuss those with the class. And there's a list of those possible observations on page 7, and all of your students, all of you are going to have students at different levels. So this may be something that they're very familiar with, or it may not be. So then if the students haven't already, they're likely coming up with the conclusion that they probably need to take a closer look at those chemicals that are coming out of the vent. And again, you'll recall the simple chemicals that were on the far left-hand side of that food web. So then you can share this illustration, and if we can keep this illustration up on the screen for just a moment, and you can guide the students through noticing the chemicals and the heavy metals that are coming out of the vent. So in this illustration, you can see the seawater seeping down through the cracks and fissures in the ocean crust, and then you can see it meeting the magma, and then the black smoker coming out, and the precipitate forming, and then and also all the different metals and chemicals floating into the water column. And you'll recall Ashton Flinder's terrific description of that fluid movement and the mineral precipitates forming the chimneys at these vent systems from the Deep Sea Dialogues video. So from there, we start considering that the students are discovering patterns, and they've used the chemical equation to identify the components of photosynthesis. So now we can look at the components of chemosynthesis, being carbon dioxide, hydrogen sulfide, and oxygen, producing the sugar, the sulfur, and the water. And as the students compare the two processes, you should hear them notice things like both the chemical equations include a compound made of carbon, hydrogen, and oxygen as a product. And some students might even notice the ratio of carbon, hydrogen, and oxygen is 1 to 1 to 1 in both chemical equations, and that these compounds represent simple sugars. So the point here then is to emphasize that both of these processes produce simple sugars, and prompt the students to share what they know or think they know about how plants and animals use simple sugar. And then prompt them to think about how bacteria use those simple sugars. And the students will likely say that the bacteria must use the sugar they produce the same way that plants use the sugar they produce in photosynthesis. And to support this particular thought process, I want to share with you a little clip from this really wonderful video by Ed Yong, and some of you may be familiar with it, and some of you may not. It's produced by I Contain Multitudes and PBS Media, and called "How Giant Tube Worms Survive at Hydrothermal Vents." This is a 10-minute video featuring Ed Yong, who is really quite humorously introducing the discovery of giant tube worms, and Dr. Colleen Cavanaugh explaining how she first identified the symbiotic relationship between the vent bacteria and giant tube worms and the process of chemosynthesis. So I'm just going to share with you a very short clip of this 10-minute video.
Plants do photosynthesis. They need water. They need water and carbon dioxide, which they transform into sugars using the energy in sunlight. But the worms can't do that.
It's dark. We're 2.5 kilometers down up to, to even deeper. That's, over 1.5 miles deep. So it's complete darkness in the deep sea.
So instead of sunlight, the bacteria ingest and process the sulfides from the vents. In doing so, they excrete sulfur, but they also release energy, which they use to make food for themselves and for the worms. And that's what chemosynthesis is, making food not with solar power, but with chemical power.
So it's apparent from a mouthless and gutless point of view that the worm is benefiting from getting its food from the bacterium. When you're a bacterium inside of the animal, and you've somehow convinced the host to provide you with the sulfide and the oxygen, then you have easy street.
I just think that's an absolutely wonderful video and depiction of the interactions that within chemosynthesis within a tube worm. And I would encourage reiterating after showing the video that the bacteria ingest and process the hydrogen sulfides from the vents. They excrete the sulfur and release the energy to make food. And they're either making that food for themselves, or they might also be making it for symbionts, like in this case, the giant tube worm. So I encourage you, if you haven't seen it, to watch that entire video. And almost finally, on page 9, we start having the students synthesize their thoughts. So how are we going to get the students to really think about what it is that they've learned, and how are we going to see what they've learned? So here we have the students create a model to illustrate their developed thinking. So they're explaining how the ecosystems exist and thrive in the absence of sunlight. And also they're going to predict what would happen if the vent became extinct. So the model suggested here is a comic strip divided into 3 sections representing a vent ecosystem over time. There's a frame 1 would be an active, thriving hydrothermal vent. Frame 2 would be a vent system that just became extinct. And frame 3 would be a vent system that's been extinct over a long period of time. And it's important to remind the students that their models should represent all of the ecosystem elements that they've learned about: the organisms, the interactions between the geology, the biology, and the chemistry, and also illustrate or explain how those components are interacting. And on that page, there's some ideas provided to you to prompt students if more information is needed. So then once all of that is complete, you can post the models or host a gallery walk and give students an opportunity to observe one another's models and then reflect and revise their models. And once they've done that, you can encourage them to use evidence or science reasoning to support their claims and their predictions. So then we have a video from Ocean Exploration Trust called Smoking Chimney and Pompeii Worms, which is from 2015 from an expedition also in the Galapagos. And in this video, there's an active hydrothermal chimney and a diverse colony of chemosynthetic ecosystems that the scientists were observing, but the vent was very near extinction. So I'll share a short clip of that with you here, just to give you a taste of the elements of that video.
Tim
Correct.
Shank says 150
Yeah,
Celsius.
it seems to be shutting down. The clams are all dead. So it seems to be in the waning period. We've found several completely dead ones as we came down the same fissure pattern. But this is at the top of what we're now calling Mount Hercules. It's a central volcano right here. And this seems to be the last gasp of this cycle.
And if you didn't recognize that voice, that is Dr. Bob Ballard from Ocean Exploration Trust, and one of the folks who was on the team that discovered the first hydrothermal vents and tube worms in 1977. So also to support that extinct concept, there is a very short log from a 2002 Galapagos Rift expedition from OER that describes how scientists went down in the Alvin submersible and came upon some vent mussels, some mussel shells that were evidence of vents, but there were no living organisms there. And then quickly moving into wrapping this up and having students determine what it is that they figured out, you can have them use their models to explain why these vent ecosystems change when the vent becomes extinct, and think about what they still have to figure out, and you can use those discussions to help guide possible further investigations. We've also provided you with a couple of optional really great videos to expand or wrap up your instruction. The Oasis of Life video is a longer version of the short clip on the Marianas Trench that we showed you in the beginning. And Ocean Exploration Trust has a great video sharing 40 years of hydrothermal vent exploration. There are some extensions that you can share, you can take a look at and read on your own, but I did want to mention one activity by Ocean Exploration Trust called Living on a Chimney. And this one uses very similar creature cards to the ones that we're sharing with you. And in this one, they expand a little bit farther in the food web food chain concept and construct a model of a food web with an emphasis on trophic levels, trophic positions, and ecological niches, and also determine spatial distribution of organisms across the vent community based on provided evidence. So I encourage you to take a look at that activity, and also note that that is offered on their website in Spanish. There is a great section on assessment that you can look at yourselves, which provides guidance for evaluating student models in relationship to pertinent standards. And lastly, the hot links on the last page, some of those are not active yet, but you do have everything that you need to do this activity in your Google folder or in the activity. And I encourage you to bring this activity and any resource questions that you might have and your thoughts to your follow-up. And if this might fit into your instruction between now and your follow-up, we'd love to hear how it works for you and also see some images of the comic strip models that your students might come up with. So I know that was a lot, and student sense making, again, is a different approach to an activity, and your feedback is more than welcomed. And with that, I'm going to turn it over to Alexandra Puritz from the National Marine Sanctuary Foundation to talk with you about resources.
Hi, everyone. First, I just want to say sorry, there's been a little bit of confusion with the Google Drive, but it sounds like everyone should have access to that now. If you don't or are still having issues with it, please let us know. So thank you, Susan. My name is Alexandra. I'm with the OER education team through the National Marine Sanctuary Foundation. I'm going to take a few minutes now right before we wrap up to show you how to access some additional relevant educational resources on ocean exploration topics on the websites of OER and our partners Ocean Exploration Trust and Schmidt Ocean Institute. Many of these links are also in the resource list that you have, but we just want to show you how to navigate the websites. So first, we're going to go to NOAA OER. Our website is oceanexplorer.noaa.gov. Again, these are in your resource list. If you hover over to the Education tab and click on Themes, yep, click on Themes. Scroll down a little bit to Vents and Volcanoes. And if you click on that, this is where we have all of our essays, lessons, multimedia, expedition info, and ocean exploration careers relevant to this topic. Just want to quickly let you know, in multimedia, you'll find all of our image and video galleries. and you'll recognize some of these from the activity that we shared with you. Also, under expeditions is where you can find all relevant information about our previous expeditions that have explored vents and volcanoes. So next, we're going to go to the Ocean Exploration Trust website. Their website is nautiluslive.org. If you click on education and then go to education resources, finding resources. On this page, you can look up resources by theme, by resource type, and also you can search for education resources by topic, by DCI, by grade level. So if we go back up to the top of this page and click on STEM learning modules, and scroll down just a little bit, you'll find Living on a Chimney. This is a really great activity by Ocean Exploration Trust that has minimal prep and the material cost is quite low. And it's a good activity for introducing students to the ecological relationships and organisms on vents. And so if you scroll down a little bit, you'll see that all of their documents are there and they're also available in Spanish. So we wanted to highlight this activity for you. Also, if you go back up to the top of the page and click on Gallery, there is a drop-down box and you can select hydrothermal vents. And when you select that, all of OET's blogs, videos, and photo albums that involve hydrothermal vents will come up. Lastly, we're going to go to the Schmidt Ocean Institute's website. Their website is schmidtocean.org. And if you click on education and click on the RV Falkor inspired lesson plans, you will see that there is a virtual vents lesson plan. It's the third one down. And this is a great lesson plan to teach about how vents are formed, vent chemistry, and biodiversity. Lastly, if we go back to Schmidt's page and click on Gallery, here you can scroll through all of their images and video and look for anything that is related to hydrothermal vents. And they have really amazing, beautiful photos and images. So if you scroll through those 5 pages, you can look for ones that are relevant to vents. Also, just want to let everybody know that when our expeditions are in action, us and our partners, we have live streams from the vessels and of both the vessels and our ROV dives. And these can be accessed directly from these websites when we have expeditions going. So it might be something really fun for you to tune into with your students. So we encourage you to explore all of these websites. They have some amazing resources with lots of information on ocean exploration. And again, many of these are linked in your resource list. And we hope that you find it useful. So thanks, and back to you, Susan.
Terrific, Alex. Thank you so much for that. We absolutely, between our 3 organizations, and other organizations, have tremendous resources out there on hydrothermal vents. So we encourage you to explore them all and essentially make up your own playlist for teaching about vents. And with that, I'd like to bring Peter back for a moment.
Hello.
Hi, Peter. Glad to have you back.
Thank you.
Yeah, I just have one last question for you, which is kind of a doozy, and there's probably not one answer. But I'm wondering about one of the most amazing experiences that you've had in your work in relationship to vents and ocean exploration.
Oh, yeah, there really are so many to pick from. One that stands out, though, is that I had an amazing experience a few years ago that involved telepresence. Now, I've been going to sea for many years, and I absolutely love it. I also recognize that there are only so many spots for people on board ship, and it's wonderful to see telepresence, the ability to use this kind of conferencing to bring lots of people to see, right? I mean, I love that. But here it was a few years ago, getting onto an airplane to fly from Boston to San Diego and feeling really bummed about not being able to participate in this one expedition. Just, I couldn't do it. And they were sampling these iron oxidizing microbes at a site that I was really excited about. Well, I got to tell you, just as soon as we reached altitude, I thought, I wonder if I can watch the Nautilus live stream from my airline seat. And lo and behold, I could. And while I couldn't talk to them, I started typing, and I'm like, hey folks, it's me, you know, is there any chance that I can watch you and we can collect one of these amazing samples? And they were wonderful about it. The folks on board ship said, yeah, let's give it a go. So somebody paid attention to the public chat, and another person was talking so I could hear what was going on. And lo and behold, I'm watching video of the seafloor from 35,000 feet, watching someone do work 8,000 feet underwater. And that was mind-blowing. I think the reason I picked that story, Susan, is because it is a reminder that technologies can and do change. And for the students out there who are listening in, you all may have exciting ways to take the technologies that you are familiar with, or you may have new ideas for new technologies and want to apply them to the study of the ocean and the world around us. So that is definitely a real zinger.
That's amazing. So you're miles in the sky and you're live exploring miles in the depths of the ocean.
So cool.
Right?
So cool.
It's so cool. It's so cool. It's as cool as the rover landing on Mars to me, right? It's just totally different environments, equally amazing.
It's true. I'll be a little cheeky and say that, the whole rover program, is a couple of $1 billion, and I got to do this for the price of an airline ticket. I'm just
Well, it's all about taking technologies and expanding it and making it as economical and as efficient as possible, right?
Well said.
Yeah. Well, Peter, again, thank you so very much. We're gonna meet with our site partners here briefly, but really appreciate your time this evening.
The pleasure is mine. Thank you all for the invitation, and thank you folks out there who are listening in. And someone let me know what eats the Yeti crabs. I'm all ears.
Thank you. So could we bring our site partners back up on the screen for a moment? Great. We have our colleague Deborah from New Mexico. Deborah, can you just repeat when your follow-up is?
Hi, everyone. Yes, the follow-up for the Albuquerque Biopark is March 10th at 4 p.m., and I will send the Zoom link out to everyone next week, early next week.
Super. Thanks, Deborah. We look forward to that. And Tina at Dauphin Island?
Yes, our follow-up is Tuesday, March 16th at 5 p.m. Central.
Okay, terrific. And Great Lakes Aquarium?
Yeah, our follow-up is on March 25th at 5 p.m. Central.
Super. Looking forward to that one too. And Loveland Living Planet Aquarium?
Ours is also on March 10th, so that's just 2 weeks away from now, and it's going to be at the same time, which was 4 o'clock Mountain Standard.
Great, thank you. And lastly, Rosie in Texas.
Our follow-up is March the 17th at 6 p.m. Central Time, and you will be getting a link from me in the next week.
Great. Thank you all for your support and your participation in this workshop. We look forward to everybody participating in the follow-ups. And participants, this is a time for you to engage in deeper conversation with your colleagues about how these materials can be used, particularly in your current teaching circumstances, which I know are quite challenging. And if you have any burning questions that didn't get answered, please feel free to send them to us using the email address that's at the bottom of the last page of your activity. And we're looking forward to offering more of these events in the future. We so appreciate your time today, and thank you for being with us. And again, a big thanks to Peter Gerges for joining us from Harvard. And thanks to our Inner Space Center colleagues behind the scenes, and the National Marine Sanctuary Foundation, and all of our OER staff who have been working hard behind the scenes. And please look for our next event later this year. And I wish you all a wonderful evening. Thanks so much.